I'm George Demopoulos at the Malaria Research Institute at the Johns Hopkins School of Public Health. And now we will show you a procedure on how to infect Anopheles gambia mosquitoes with Plasmodium falciparum parasites and how to determine the infection phenotype on the mosquito midgut. For this procedure, the basic equipment needed are: we need a CO2 incubator for the parasite culture. Alternatively, if you don't have a CO2 incubator, you can use a candle jar. We also need an insectary, of course, standard molecular biology and cell biology equipment such as dissection microscopes, light microscopes. We need water baths. We need a mosquito membrane feeder system and centrifuges.
Hi, my name is Shuchishmita Das and I'm in the lab of Dr. George Demopoulos. I'm a postdoc.
My name is Lindsay Garver and I'm a PhD student.
The first step of the Plasmodium infection is to have the Plasmodium culture ready. If you don't have a carbon dioxide incubator, then you can have a candle jar where you can put the 6-well plate having the gametophyte culture, then light the candle and then seal the jar. The first step is to have the fresh human blood and serum and put them at 37 in a water bath, first to thaw and also to have them like warm, because you need really warm blood and warm serum. To prepare the blood, you need to aliquot 1 ml of the fresh human blood in an ependoff. The fresh blood has to be spun at 2000 rpm for 5 minutes to pellet down the erythrocytes. After the spin is over, the clear soup has to be discarded and the red pellets, which is the RBC, needs to be washed 3 times in human sera. We add equal volume of human sera to the pellet, mix them and spin again at 2000 rpm for 5 minutes. This step is repeated 3 times. Finally, to have a red blood cell pellets ready to be added to the parasite culture. Okay. The Plasmodium culture, which is in the CO2 incubator, it has to be maintained in a RPMI medium for 16 days, and on day 16 they are ready for the infection. Since we are working with the Plasmodium culture, care must be taken at every steps. The first step is that the parasites should be grown in a BCL2 lab, and it should be performed in a hood. All the pipette tips and all other ependoffs, everything has to be discarded in a biohazard box, and any other solutions has to be like, or pipette man, they have to be washed in 10% bleach, and you have to wear gloves, lab coats. From the day 16 culture, you need to take a small drop of blood on the glass slide, make a smear and do a Ginsa stain, and under the light microscope we can determine the gametocytemia percentage. From the 16-day-old gametocyte culture, remove most of the media, taking care not to disturb or suck up any parasites, which is usually at the bottom of the well. Pipette the rest of the media and parasites, mixing the parasites in the red blood cells into an ependoff. The parasites, which is in the ependoff, is then put in a centrifuge to spin at 2000 rpm for 5 minutes. After the spin, the parasites will pellet with the red blood cells. The supernatant is the RPMI media, and at the bottom the pellet has the parasites with the RBC. Remove the soup, which is the RPMI media. Care should be taken not to disturb the parasite and the red blood cells. The gametocytemia percentage, which is determined by the Ginsa stain, is usually in the range of 2 to 4%. Then when the red blood cells and the parasites are ready, after the spin, they are mixed together, and the volume of blood that needs to be added is determined by the final gametocytemia percentage which needs to be achieved, and it's usually in the range of 0.3 to 1%. And this is what will be used for the feeding of the Anopheles mosquitoes for the Plasmodium falciparum infection. This is a glass feeder which we use for the membrane feeding for the mosquitoes. We put the blood inside through the small hole that you see, and it is contained in that at the bottom. On the side are two nozzles which are connected to a water bath which is maintained at 37 degrees. To prepare the feeder for the feeding, we take a small piece of parafilm, we stretch from the two ends from all the four sides, and then put that on the open side of the glass feeder so as to make a thin sheet of membrane, just as to mimic the skin. This is what is called the membrane feeding. This is a circulating water bath which maintains the water at 37 and also circulates the water through the glass feeder so that the blood is maintained warm at 37. So we connect the glass feeder through the small capillaries and connect that to the circulating water bath and turn the water bath on so that the water circulates, and the water and blood, they never mix as they are in two separate chambers. This is a small cup containing the Anopheles gambiae mosquitoes. To begin with, the glass feeder is put on the cage and a weight is put on the glass feeder so that the membrane, which is the parafilm, is in flush with the mesh net. Around 200 microliter of the blood, which contains the parasites with the required gametocytemia percentage, are added to the feeder, making sure that the blood goes all the way down and the mosquitoes can get access to the blood. The feeding is continued for around 30 minutes. Once the feeding is complete, the glass feeder is washed thoroughly in 10% bleach, and the rest of the membranes and tips with ependovs, they are discarded in biohazard box. After the blood feeding, the unfed mosquitoes have to be removed from the fed ones. To do this, we knock down the mosquitoes by putting the small cups containing the blood-fed mosquitoes in cold room for 5 to 10 minutes. As we can see, the mosquitoes are already down in the cold. We open the lid and transfer the mosquitoes in a small petri dish which is kept on an ice bucket so that the mosquitoes are kept in cold all through and they cannot escape. Remember, these are Plasmodium infected mosquitoes, so all care must be taken not to let them go loose. Once the mosquitoes are down in the cold room, we transfer the mosquitoes to the normal insectary which is maintained at the room temperature, but we put the mosquitoes always in the petri dish which is kept in the ice bucket so they are there, so that they are always down. You can see how the fed mosquitoes can be removed from the unfed ones. The fed ones have the blood in them, they are fully engorged. Some of them have fed on blood partially, so they are not fully engorged. They have a small blood in the abdomen, but others have a lot of blood in their abdomen. The unfed ones do not have any blood in their abdomen, and so you can easily distinguish between the fed and the unfed ones. Shown here are the adult female mosquitoes which have fed on the blood. So we will transfer these mosquitoes from the petri dish to the wax-lined cardboard cups. After putting the blood-fed mosquitoes in the cups, we put a small piece of cotton soaked in 10% sucrose so that they can feed on the 10% sucrose for the next 7 days that you are going to incubate, and also put a small folded wet paper towel so as to maintain the moisture within the cup and cover with a petri dish. The Plasmodium infected mosquitoes are put in another small cage so it has to have a double protection. And we'll put the cage in a Bill-3 lab, which is maintained at 27 degrees centigrade and 80% humidity. 7 days after the blood feeding, the mosquitoes which were kept in the insectary in the double caged box, they were taken out from the insectary and taken to the cold room to knock the mosquitoes down. After the mosquitoes are down, they are transferred to a petri dish which is kept on an ice bucket. Now we are getting ready to dissect the mosquitoes. We'll dissect the midgut and look for the oocyst. The first step is to add 100 microliter of PBS on a glass slide mounted on a dissection microscope. The next step is using a fine tip forceps. We transfer a single mosquito from the petri dish to the PBS, which is on the glass slide mounted on the dissection microscope. The others are still left on the chilled petri dish. To dissect the mosquitoes, hold the mosquitoes at the thorax with one of the forceps and the posterior abdomen with another forceps. Then pull the body part slowly until the midgut is exposed. It looks like a white sac-like body. Use forceps to remove the excess tissue and the other body parts. Yeah, this is the midgut, which looks like a white sac-like body, which has the oocysts. We'll transfer this midgut now to a 12-well glass slide and immerse in a 5 to 10 microliter of 0.2% mercurochrome, which will stain the midgut. We cover the slide with a cover slip. We do this step for all the infected mosquitoes. The gut looks like a bright pink, and we'll transfer this slide to a light microscope to count the oocyst load. We are now looking at the midguts dissected from the infected mosquitoes. The midgut look like a sac-like body, and with the mercurochrome stain, they have a very slight pinkish color, and the oocyst should have a bright pink against the faint pink gut tissue. These midguts, though they are from the infected mosquitoes, but they do not show any oocyst, so they may be not infected. But if a midgut would have been infected, the oocyst would have shown as a small bright pink circle, and there can be numerous, like between 20 to 500 sometimes within one single midgut.
This procedure involved infecting human blood with Plasmodium falciparum parasites, feeding that infected blood to Anopheles gambiae mosquitoes, dissecting the midguts out of those mosquitoes, and determining the oocyst counts within those infected midguts. This technique is important because it is the closest approximation of human malaria transmission that we can replicate in the laboratory.